Inside a living cell, DNA replication relies on an intricate machinery of specialized enzymes. In a laboratory, the polymerase chain reaction, or PCR, strips that complexity away. Instead of using unwinding enzymes like helicase to open the double helix, PCR relies on sheer thermodynamic force to pry the strands apart. Starting with just a handful of target sequences, that exponential geometry generates millions, and eventually billions, of exact copies within a few hours. Substituting biological complexity with controlled thermal cycling yields an exceptionally powerful amplification engine, one that serves as the basis for modern molecular diagnostics. We use this theoretical power to search for microscopic traces of biological material, like detecting a viral pathogen in a patient's blood sample. But this exponential system possesses a fatal kinetic flaw. It chokes on its own success, crashing into a physical ceiling known as the kinetic plateau. When the reaction hits this plateau, the linear mathematical relationship between the final amount of DNA and the original starting sample is entirely destroyed. Because that linearity is lost, reading the reaction at its endpoint makes it mathematically impossible to determine how much genetic material you started with. To turn PCR into a high-resolution tool capable of measuring exact viral loads, engineers had to deconstruct the thermal cycle and build variants designed specifically to bypass that plateau. A single PCR cycle moves through 3 strict thermodynamic phases. Step 1 is denaturation, requiring extreme heat between 94 and 98 degrees Celsius. That specific thermal energy breaks the weak hydrogen bonds holding the base pairs together, forcing the double-stranded DNA to separate into single strands. Step 2 is annealing. The temperature drops to between 50 and 65 degrees Celsius, allowing short, synthetic DNA fragments called oligonucleotide primers to bind to the specific target region we want to copy. This vector model illustrates that binding process. The exact temperature required here is strictly dictated by the primer's melting temperature, which is determined by its length and base composition. Mismatched annealing temperatures cause system failures, like primers binding to each other to form useless primer dimers. But if correct, we move to extension. The system heats to 72 degrees, and Taq-Dena'ine polymerase locks onto the bound primers, synthesizing the new complementary strand. This enzyme cannot function alone. It requires magnesium ions to act as a vital structural cofactor, stabilizing the reaction as the new nucleotides are added. Completing these 3 steps, denature, anneal, and extend, exactly doubles the target DNA, resetting the system for the next cycle of the exponential pump. We map this on a Cartesian graph, cycle number versus DNA quantity. Ideally, this plots an unimpeded exponential curve, but physical reality cannot sustain infinite math. Running out of reagents, the reaction loses momentum, flattening into a hard plateau around cycle 30. This depletion is compounded by secondary failures, including product inhibition from massive DNA crowding and the gradual heat inactivation of the Taq polymerase after repeated exposures to 94-degree temperatures. We can see the clinical consequence of this by plotting 2 hypothetical patient samples, one with a massively high viral load and one with barely any virus present at all. If you run both samples through a standard PCR protocol, the exponential math forces both starting quantities to ultimately crash into that exact same physical plateau limit. When a technician runs an endpoint readout, like an agarose gel, the bands for both patients look completely identical, masking the massive discrepancy in initial disease states. The kinetic plateau renders endpoint PCR an excellent qualitative tool for answering if a pathogen is present, but an utterly useless quantitative tool for determining how much of it is actually there. The logical engineering solution to this quantification problem is a temporal one. We have to measure the DNA concentration before the reaction exhausts itself at the plateau. In real-time PCR, or qPCR, we introduce fluorescent probes to actively measure amplification. We establish a horizontal cycle threshold, or CT line, well below the plateau. The CT value is the exact moment a sample's fluorescence crosses that threshold, revealing distinct curves. This creates a direct inverse mathematical relationship. A low CT value indicates a high initial viral load, because a larger starting sample will generate enough copies to cross the threshold in very few cycles. Conversely, A high CT value means the machine had to run many more amplification cycles before the signal became detectable, pointing to a very low initial viral load. By taking these CT values and plotting them against a known standard curve, technicians extract an absolute, highly accurate count of the initial DNA copies per milliliter. qPCR solves the quantification problem temporally. By reading the data early, it rescues the clinical utility of PCR, allowing physicians to actively monitor disease progression and adjust antiviral therapies. qPCR is highly effective, but its temporal solution carries one strict limitation. It relies entirely on comparing your sample to an established standard curve to calculate those numbers. In certain cutting-edge clinical scenarios, standard curves are impractical or impossible to generate, such as attempting to count microscopic trace amounts of a rare cancer mutation from a liquid blood biopsy. To solve this, bioengineers developed digital PCR. It abandons the temporal measurement model entirely, opting instead for a purely spatial solution. In digital PCR, we take a single reaction volume and partition it into a spatial grid of tens of thousands of microscopic droplets. Applying the thermal cycle yields a binary readout. Droplets containing target DNA light up as positive ones. Empty droplets remain dark zeros. Technicians then apply Poisson statistics to the exact ratio of positive to negative droplets, allowing them to precisely count the absolute number of mutant alleles present in the sample. By spatially isolating the reaction into thousands of microchambers, dPCR effectively digitizes the amplification process. It delivers absolute quantification with incredible sensitivity without ever needing a standard curve. The power of this core thermodynamic engine is that we can adapt it for highly specific requirements across different scientific domains, simply by altering the initial inputs. In clinical diagnostics, we use RT-PCR to detect RNA pathogens like SARS-CoV-2. As we see in this sample preparation, this requires a mandatory pre-step using reverse transcriptase enzymes to convert the viral RNA into complementary DNA before the thermal cycle can even begin. In other applications, we use multiplex PCR. By adding multiple different primer sets into a single reaction tube, we can amplify several distinct genetic targets simultaneously. Forensic scientists rely heavily on this multiplexing capability. They use it to simultaneously amplify highly polymorphic short tandem repeats from heavily degraded trace evidence left at a crime scene. PCR's immense value across both biotechnology and forensic science is a direct result of this adaptability, which is always anchored by the strict sequence specificity dictated by those initial primers. The evolution of this technology takes us from the basic qualitative limitations of standard endpoint PCR, all the way to the high-resolution diagnostic power of its modern variants. That entire evolutionary track was forced by a strict physical reality, the unavoidable kinetic plateau where the reaction ultimately consumes itself. Mapping the mechanics of that limitation allowed molecular biologists to engineer workarounds, leading to the real-time temporal measurement of qPCR and the high-resolution spatial counting of digital PCR. The modern outputs look drastically different, ranging from simple forensic electrophorangram peaks to thousands of digitized, illuminated droplet grids. But regardless of exactly how advanced the clinical readout becomes, the underlying biological substitute remains a brilliantly simple thermodynamic engine. Heat to separate, prime to target, extend to copy, repeat.